Today we're going to be talking about DNA. We're going to have a brief overview of this very important molecule. Now, some of you probably have a pretty extensive background in DNA, what it is and what it does in the cell, and why it's important to living things. But some of you may be a little rusty on the subject and maybe haven't had any classes that discuss DNA since high school. So our goal for today is to get everybody on the same footing so that when we move on to our next topic, which is transcription, we'll all be on the same level. Now, before we can talk about DNA in detail, we need to talk about the central dogma. The central dogma is simply a set of statements that discusses how information flows within a cell. Those statements are that DNA is transcribed into RNA, that RNA is translated into protein, and that protein creates traits. When I use that word trait, what I'm talking about is a characteristic, something that you could point to and talk about as a characteristic of an organism. It might be something physical, it might be something behavioral, it might be something biochemical. All of those things are characteristics, and all of those things are traits. So in this slide, we can see a diagram of this flow of information from DNA to RNA to protein. The process where DNA is used to make RNA is called transcription, and the process where RNA is used to make protein is called translation. We say that DNA is transcribed to RNA, and RNA is translated into protein. Now, DNA, like several other macromolecules, can be described on certain levels of chemical structure. When we talk about chemical structure in this way, we can talk about three categories or levels of complexity of structure: primary, secondary, and tertiary. Primary structure is just a description of the simplest way a molecule can be described. Secondary structure includes usually some folding or some shape development in the chemical. So this is the next level of complexity. And finally, tertiary or third order chemical structure is typically the fully mature three-dimensional form. Tends to be folded and tends to be quite complex. So these terms, primary, secondary, and tertiary, are generic terms for describing the levels of complexity of structure of any macromolecule, including DNA. So DNA, remember, is actually an abbreviation for the chemical deoxyribonucleic acid. The primary structure of DNA, the way we could describe it in its simplest form, is just a strand of deoxyribonucleotides, or the monomer of DNA, that are put into a basic sequence. So DNA, if you remember, is actually an abbreviation for the molecule deoxyribonucleic acid. And when we talk about DNA in its primary or its simplest structure, we're talking about a strand of monomers. Monomers that, when they're put together into a strand, produce a polymer. The monomer of DNA is a deoxyribonucleotide, and that's what you can see in the bottom part of this slide. Now, when we bind one deoxyribonucleotide to another and another and another, we create this strand or this polymer that is DNA. So what makes each piece of DNA unique is the specific sequence that those individual deoxyribonucleotides are in. So let's take a look at the bottom of the slide here, and you'll see an individual deoxyribonucleotide. This has three basic components to it, and these are the three components I'd like you to learn. There's a sugar or a carbohydrate component, and that's shown here in the rose color. There's a phosphate group attached to one of the carbons, and there's what we call a base. This is a component of DNA that can either be adenine, thymine, guanine, or cytosine. Now you'll often hear abbreviations used for these bases. A for adenine, T for thymine, G for guanine, and C for cytosine. And I'll be using those abbreviations in class as we talk about DNA. Now, one of the things you'll notice on this slide is that there are numbers next to a couple of these carbons. There's a 3' number for this carbon at this location, and there's a 5' number for this carbon at this location. In a molecule like DNA, each carbon gets a number. This would be carbon number 1. This is carbon number 2. This is carbon number 3, number 4, and number 5. The reason the 3' and the 5' carbons are highlighted in the slide is because they play an important role in joining one deoxyribonucleotide to the next to build a strand of DNA in the primary structure. So, in this slide we can see a short molecule of DNA, and all it is a strand of bases in a specific sequence. And in this case, the bases are adenine here, guanine here, cytosine here, and thymine at the bottom. And the way I can tell each base apart is because each base has a different structure. Now you'll notice the top two bases, the adenine and the guanine, look very similar. They have a two-ring structure. Whereas the last two, the cytosine and the thymine, are single-ring structures. The adenine and guanine belong to a group of molecules called purines, and the cytosine and thymine belong to a group of molecules called pyrimidines. So these are the purine bases, and these are the pyrimidine bases. Now, I don't expect you to learn the structure of the bases, but I do want you to know the names of the 4 of them, and I want you to know which ones are purines and which ones are pyrimidines. Now, in this slide, we've taken the next step in that we've taken one strand of DNA and we've joined it together with another strand of DNA. So we have a 2-stranded DNA molecule, and as it turns out, in nature, DNA never occurs in a single strand. It's always double-stranded. So what we can see in this picture is that the bases join together, not with covalent bonds, but with hydrogen bonds. You can see the guanine and the cytosine have 3 hydrogen bonds between them, and the adenine and the thymine have 2. So remember, hydrogen bonds are not strong bonds, they're attractions, but they are responsible for holding the 2 strands of DNA together in the very classical double helix. The other thing you can see in this slide is that for each of the individual monomers, each of the deoxyribonucleotides, you can't see the sugar and you can't see the phosphate group. And what usually happens when we draw DNA in this way is we eliminate the sugar and the phosphate, and instead we draw a thick line like you see here in red. This is what we call the sugar-phosphate backbone. So this red line represents 2 individual sugars, 2 individual phosphates, and all we can see are the bases that are jutting off of each monomer. Same thing on the other side. Here's my sugar-phosphate backbone, and here are the bases jutting off. Now you'll notice that on either side of this double-stranded DNA, the sugar-phosphate backbones are oriented in a different direction. The 5' carbon is up here, and the 3' carbon is down here, along with the arrow head. And on the other side, the 5' carbon is here, and the 3' carbon is here, along with the arrowhead. So DNA has directionality. And one thing to remember is that we can only add new bases onto DNA when we are synthesizing or creating DNA. We can only add new bases onto the 3' end. So if this molecule were to get any longer with the addition of more bases or more deoxyribonucleotides, they would be added on here on this side and here on this side. So we talk about DNA a fair amount in our culture, and we usually talk about it with regards to genes. Most people in our culture use that word gene with limited understanding, though. They don't understand what a gene is, although they do understand that it can impact a person in terms of their traits and in terms of their health. So we need a formal definition for gene. And the one that we're going to use has 3 parts to it. First of all, a gene is a length of DNA that's in the primary sequence. And remember, all that means is that there's a specific order to the bases in that length of DNA. We're going to say that a gene encodes the information that's necessary for building a protein or a polypeptide, and I'll explain the difference between those two terms when we go into our protein lecture. So most people in our culture use the term DNA relatively freely. They also use the term gene pretty freely, although a lot of people don't really have an understanding of what a gene is. Now we're going to have a formal definition for this term gene, and it has 3 parts. First of all, a gene is a length of DNA that's in the primary sequence. And remember, all that means is that it's a strand of bases joined together in a specific order. A gene also encodes information for building a polypeptide or a protein. And finally, a gene contains regulatory information, a sequence of bases that help determine when, and in fact if, it gets transcribed and translated into a protein. Now, humans and many other organisms have 2 copies of every gene they have in all of their cells. They inherited 1 copy from their mother and 1 copy from their father. Each version of these genes is called an allele. So you have 2 copies for every gene in your DNA. And all an allele is a particular version or a form of a gene. Now, some of you may have heard of the Human Genome Project, and I only wanted to mention it here because we have some very important conclusions from the Human Genome Project that are important to us in biology. The Human Genome Project was a massive undertaking which was designed to determine the order of the bases, the primary structure of the entire molecule of DNA that's inside a human being. As you can imagine, it took a lot of time and a lot of money, and it was largely government funded. But we now know that there are 3 billion base pairs in the human DNA molecule. Now you'll notice I say base pairs and not bases, because remember, in nature, DNA doesn't occur as a single strand. It always occurs as a double-stranded molecule. So 3 billion base pairs is a tremendous number of base pairs. Hard for us to even comprehend. There are so many. And you might ask yourself, well, if there are 3 billion base pairs, how many genes are there inside human DNA? And the answer is we don't really know yet. We're still discovering genes every day. And we're also discovering that there are pieces of DNA and lengths of DNA that are not genes in the human DNA molecule. We don't know yet what all of these sequences do. We're still learning. But right now we can make a pretty decent estimate that there are between 20 and 25,000 genes inside a molecule of human DNA. So now that we know the primary structure of DNA, that specific order to the bases in a strand of DNA, we can talk about secondary structure. And remember, all this term refers to is the next level of complexity of a molecule of DNA. And in the case of DNA, this next level of complexity is simply the double helix. Remember, we said DNA never occurs as a single strand out in nature. It always binds two strands together. And the binding occurs with hydrogen bonds between the bases of the two strands. Now, in this diagram, you can see that everywhere there's a G on one strand, there's a C on the other strand. Those two bases are complementary. And everywhere there's an A on one strand, there's a T on the other strand, because those bases are complementary. So you end up building, in essence, a ladder with a sugar phosphate backbone here and bases jutting off, and a sugar phosphate backbone here and bases jutting off. This ladder doesn't stay like this, though. It doesn't stay in that sort of flat formation. Instead, it assumes a three-dimensional helical form, and this is why we refer to it as a double helix. Now, there is a third order or tertiary structure to DNA, and that is called the chromosome. All a chromosome is a DNA-containing structure, or if you like, a gene-carrying structure that consists of a molecule of DNA and associated proteins called histone proteins. And the purpose of histone proteins is to help make the DNA more compact. The DNA is actually going to wind around histone proteins, and those wound regions of DNA are then going to stack and pack in together inside the cell nucleus. So in this picture, you can see that happening. The bluish gray molecule here represents the double helix of DNA. And as you can see, these red sort of spherical objects, these are histone proteins. So the DNA comes down and it wraps twice around each histone protein. And then an additional histone protein, shown in yellow, helps secure those two strands to that red histone protein. Then the DNA continues on and wraps twice around the next histone, gets secured. Then it goes on, wraps twice around the next histone, gets secured, and so on, until the entire molecule of DNA becomes wrapped around histone proteins, and it's ready to be compacted into a smaller space called a chromosome. So here at the top of this slide, we can see DNA in its secondary structure, the double helix, and they're showing us how this double helix wraps around the histone proteins, as you can see here. And then those histone DNA structures start to stack one on top of the other. And then the DNA gets compacted into a much smaller area. Now you might ask yourself, why would the cell take the time and the trouble and the energy to do this with its DNA? You have to remember that DNA is arguably the most important molecule in the cell. It contains all of the information to make a new organism, and it needs to be protected. That's why the DNA gets placed into a nucleus, so it's segregated from the rest of the cell. It's also why the DNA gets wrapped around histone proteins and packed together in this way. We believe that the chromosome structure serves the purpose of protecting the DNA because it's so coiled up and it's not exposed as it would be if it were left in a linear form. The other thing to remember is that That DNA molecule, at least in humans, is 3 billion base pairs long, and you would be hard-pressed to get that molecule into a cell's nucleus unless you did something to put it into a smaller area. Now, I like this diagram because it puts everything we've said so far into one place. Again, down at the bottom of the slide, you can see this ladder formation, the basic idea of the secondary structure of DNA. Then it twists into a double helix here. We start to see it being wrapped around histone proteins here. Those histones are then stacked into a structure that's much tighter and more compact than the original DNA molecule. And that structure then coils up and gets compacted even more into what we call the chromosome. And as you can see here in this image, the chromosome is drawn as an X. That represents 2 chromosomes joined together. You inherit one of these chromosomes from each of your parents, because remember, you get 2 copies of every gene that then sits in the nucleus of the cell and contains all of the information that that cell would need to make another organism.